Thank you for the kind introduction. It is my pleasure to present today a summary of SARS-CoV-2 virology, immunology, and the diagnostic tools available to identify and track infections. In particular, we will focus on the assays and instrumentation that Bio-Rad has created and continues to develop as we increase our knowledge of COVID-19. I am an employee of Bio-Rad, however, have not received compensation for this presentation. It is intended for educational use to provide participants with scientific and evidence-based information. Today's presentation will be divided into three major sections. We will first quickly review the basics of SARS-CoV-2 infection and the body's response. We will then move on to epidemiology and dive into some of the latest statistics before covering how we diagnose infection. The remainder of the talk will focus on the unique offerings that Bio-Rad has commercialized and is developing to assist in the management of the pandemic. When SARS-CoV-2 was initially discovered, it was named the 2019 novel coronavirus and then later renamed Severe Acute Respiratory Syndrome Coronavirus 2. Infection with this virus may result in the disease known as Coronavirus Disease 2019, COVID-19. It is a new member of the coronavirus family. Coronaviruses are large, approximately 30 kb enveloped, positive sense single-stranded RNA viruses sized 80 to 220 nanometers in diameter. The envelope bears crown-like 20 nanometer in length spikes that resemble the corona of the sun under electron microscopy, hence given its name coronavirus. You can see this in the small image to the upper right of the slide. On the right, you can also see a cartoon representation of a coronavirus particle. The coronavirus particles are organized with long RNA polymers tightly packed into the center of the particle, surrounded by a protective protein capsid. In coronavirus, these proteins are called nucleocapsid. The coronavirus core particle is further surrounded by an outer lipid membrane envelope with proteins inserted. These membranes derive from the cells in which the virus was last assembled, but are modified to contain specific viral proteins, including the spike, membrane, and envelope proteins. In the lower right image, you can see a close-up view of the spike protein X-ray crystal structure with the ACE2 receptor and RBD domains indicated in yellow and blue. These are two of the three common targets for antibody assays, along with the nucleocapsid protein. Coronaviruses are zoonotic, meaning they are transmitted between animals and people. Mammals may serve as intermediate hosts, facilitating recombination and mutation events with expansion of genetic diversity. Not all coronaviruses are pandemic strains. 4 common human coronaviruses are responsible for approximately 5 to 10% of all upper respiratory and lower respiratory tract infections. 2 previous outbreaks have occurred from coronaviruses. This includes the Severe Acute Respiratory Syndrome, SARS-CoV, in 2002 in China, and the Middle East Respiratory Syndrome, MERS-CoV, that happened in 2012 in Saudi Arabia. SARS-CoV-2 was initially isolated in the BAL fluid of 3 COVID-19 patients from the Wuhan area of China in December of 2019. SARS-CoV-2 replication. Here we see the life cycle of highly pathogenic human coronaviruses such as SARS-CoV-2. SARS-CoV-2 enters host cells by first binding to the cellular receptor, angiotensin converting enzyme 2, ACE2, on the membranes of host cells expressing the receptor, such as pneumocytes and enterocytes via the surface spike protein. The spike protein then mediates virus cell membrane fusion and viral entry. Viral genomic RNA is released and translated into viral polymerase proteins. The negative sense genomic RNA is synthesized and uses a template to form subgenomic or genomic positive sense RNA. Viral RNA and nucleocapsid structural protein are replicated, transcribed, or synthesized in the cytoplasm, whereas other viral structural proteins, including S, membrane, and envelope, are transcribed and translated in the endoplasmic reticulum and transported to the Golgi. The viral N-complex and S, M, and E proteins are further assembled in the ER-Golgi intermediate compartment to form a mature virion, then released from host cells. Our knowledge of the immune response is growing and changing each week during this pandemic. Adaptive immunity is perhaps the most well-studied and involves our B and T cell response, and I'll cover that on the next two slides. For innate immunity, we know that the cellular response involves recognition of virus by binding to immunogenic antigens. This triggers signaling cascades leading to production of pro-inflammatory cytokines. These can occur in macrophages, which secrete inflammatory cytokines in the lung, as well as alveolar epithelial cells make a contribution. Neutrophils also function in this way, and we see a higher frequency in the blood of infected patients, and this may predict COVID-19 outcomes. Along with this, there are several areas of active research being explored. These include the activation of innate immune sensor TLR5, the response of killer cell immunoglobulin-like receptor polymorphisms, the presence of human leukocyte antigen, HLA polymorphisms. These have been more well studied and includes a specific allele with higher frequency associated with severe cases in a Taiwanese group. This is the HLA-B4601 allele. And then the HLA-DRB1-1202 and HLA-B0703 have been shown to be associated with increased susceptibility in Vietnam and Hong Kong, respectively. This data was published just recently in the International Journal of Immunogenetics. Humoral immunity is, of course, well understood, and the basics apply to SARS-CoV-2 as well. An antigen that binds to the B cell antigen receptor signals B cells and is at the same time internalized and processed into peptides that activate armed helper T cells. Signals from the bound antigen and from the helper T cell induce the B cell to proliferate and differentiate into a plasma cell secreting specific antibodies, shown on the top two panels on the right. These antibodies protect the host from infection in three main ways: neutralization, opsonization, and complement activation. The dynamics of the antibody secretion in COVID-19 patients is under very broad investigation. However, the correlation with protective immunity still remains unknown. Seroconversion is clear at approximately day 18, with the maximum antivirus IgG titer measured 20 days after disease onset. The picture of overall prevalence is evolving, but still unknown. However, recent data is indicating a higher prevalence of infection than indicated by standard RNA testing. As I mentioned on the last slide, there are now several pieces of data that indicate the prevalence of SARS-CoV-2 infection is much higher than indicated by viral RNA testing. In addition to several single-site or regional studies, a new cross-sectional serological study was performed by the United States CDC on a sample of residual sera obtained from persons of all ages from 10 sites across the United States. After testing greater than 16,000 samples, for most sites, it is likely that greater than 10 times, with a range of 6 to 24, more SARS-CoV-2 infections occurred than the number of reported COVID-19 cases as tested by RNA diagnostic methodology. We can see in the figure here the different cities that were tested and the different dates with the spread of the population reactive for SARS-CoV-2 antibodies. T cells play a large role in long-term immunity through recognition of the virus, and this role is actively being investigated and debated. It appears that in those with advanced disease, the T cells may no longer be fully functional, including an induction of immune cell apoptosis, especially lymphocytes and T cell populations, the triggering of the cytokine storm, impairments of antigen presenting pathway and non-optimal T cell activation, and changing of T cells from immune activated to immune exhausted type. A recent report in Lancet Respiratory Medicine studied peripheral blood from critically ill COVID-19 patients by flow cytometric analysis. The figure to the right shows the phenotypic analysis of circulating T cells. In the top left panel shows that both CD4 and CD8 T cells were hyperactivated, as reported by HLA, DR, and CD38 with substantially reduced counts. Panel B shows the higher frequency of Th17, which are both CD4 plus and CCR6 plus subset among CD4 T cells detected by flow cytometry. Lastly, panel C in the bottom right shows expression of cytotoxic granules as marked by perforin and granulicin in CD8 T cells. The results imply that overactivation of T cells manifested by increase of Th17 and high cytotoxicity of CD8 T cells accounts for, in part, the severe immune injury in these patients. Now let's talk about the epidemiology of COVID-19. The source of COVID-19 infection are the COVID-19 patients and or those who have tested positive for SARS-CoV-2 RNA, as we know that asymptomatic spread is possible. Further, recovered patients may also show positive viral load results, and the ability for them to transmit is being investigated. Most cases are mild and recovery occurs in 1 to 2 weeks. However, certain populations are at greater risk, including older people, that is people over 60 years old, and those with underlying medical conditions, such as cardiovascular disease, diabetes, chronic respiratory disease, and cancer. Patients with mild disease tend to have negative RT-PCR results at day 10, while those that have severe disease have RT-PCR positive results beyond day 10. The most common clinical features include fever, cough, malaise, and fatigue, with sputum production, headache, hemoptysis, and diarrhea being less common. This is a highly transmittable virus with an R0 of 2.2 in some populations, though CDC has settled on 2.5. For reference, seasonal flu has an R0 of 1.3, Ebola has a R0 of approximately 2, and MERS approximately 1. The routes of transmission are primarily respiratory droplets and contact transmission, though recent reports indicate presence in urine and stool of confirmed patients, leading to risk of fecal-oral transmission. Stopping the spread is important, and avoid prolonged and future outbreaks involves similar personal measures to what we have already been implementing, such as mask, hand washing, social distancing, along with ample testing and contact tracing. This is the nextstrain.org database that shows the genomic epidemiology of SARS-CoV-2, currently based on greater than 3,500 sequenced genomes. It shows that SARS-CoV-2 spread quickly around the world after it emerged in China sometime between October and December of last year. While the first 10 genomic sequences from the 9 COVID-19 patients had 99.98% sequence identity, the virus was and is mutating, as normally happens with viruses, and that a large proportion of the global genetic diversity of the virus causing COVID-19 was found in all of the hardest hit countries. Recent large-scale genetic analysis indicates that it is one virus with greater than 200 substitution sites among its 8 coding regions. Sequence and evolutionary tree analysis revealed it to be a beta coronavirus sharing 79.5% and 50% identity with SARS-CoV and MERS-CoV, respectively. This is the Johns Hopkins COVID-19 dashboard, which shows the most recent COVID-19 global cases. This was accessed in early August, so clearly this data has changed by today. But just at an overview perspective, it shows that greater than 18 million people have been infected with SARS-CoV-2, resulting in almost 700,000 deaths, with the United States accounting for approximately 26% of infections and 22% of all the deaths. This is a graph showing the time course of response and diagnosis. We're all familiar with these kind of graphs that highlight the rise and fall of various markers that can be tested using routine laboratory methods. SARS-CoV-2 RNA detection by RT-PCR is the gold standard for diagnosis, and its use is widespread. Depending on the sensitivity of the test, the presence of RNA is detectable within days of infection and possibly prior to the presence of symptoms, with peak viral load at approximately 7 to 8 days after infection. The antibody response then trails RNA, with IgM/total antibody detectable at approximately day 5, and IgG detectable at approximately day 10, though there are significant overlap and differences seen dependent on the test performed. Viral load begins to decline and nears the limits of detection after around 2 to 3 weeks. At this time, antibody remains detectable for significant periods greater than 35 days, though some reports have indicated the disappearance of antibody titers, and this is an active area of investigation. With all of this data in mind, what can we do to contain the spread and decrease mortality? From a laboratory standpoint, we need to continue to focus on 3 main areas: surveillance, screening, and diagnosis. Surveillance includes ongoing, systematic activities used to monitor for an occurrence and characterize the occurrence once detected, such as looking at the incidence and prevalence of the occurrence at a population level. Screening for COVID-19 is looking for occurrence at the individual level, even if there is no reason to suspect infection, such as known exposure. Diagnosis is what we're all mostly familiar with and is used to detect infection in individual patients suspected of COVID-19 by their healthcare provider, such as in symptomatic individuals. The remainder of this presentation will focus on the tools that are currently being used for diagnosis and monitoring, such as molecular diagnostic assays for RNA detection and serological assays for antibody detection. At the state and federal level, there is guidance available about how to make best use of the available testing capacity. This often takes the form of a tiered approach. On this slide is shown an example of the tiered approach employed in California. Tiers 1 and 2 are focused on symptomatic individuals or emergency/healthcare workers. Tiers 3 and 4 focus more on the general asymptomatic population who may have close contacts with possibly infected individuals. California then employs this tiered prioritization to enable who is able to go to their healthcare provider and receive a test at this time, with not all people in tiers 3 and 4 able to get a test dependent on the current capacity. Bio-Rad offers a suite of assays and instrumentation for SARS-CoV-2 detection. On the molecular side, this includes real-time PCR instrumentation, such as our CFX product line, as well as standards, reagents, and plastics. We also have our QX Droplet digital PCR instrumentation that has a EUA SARS-CoV-2 assay. Lastly, we offer quality controls under Exact Diagnostics that validate the entire process of a molecular assay, including extraction, amplification, and detection. On the post-infection side of the spectrum, we offer serological testing. This includes the Platelia EUA SARS-CoV-2 total antibody test and the Bioplex 2200 IgG panel that is currently in development, and I will describe both of these later on. We also offer instrumentation and quality controls. The quality controls are known as the VeriTrol reactive control and the VeriClear non-reactive control. Diagnosis mainly relies on RT-PCR testing of respiratory specimens. This involves obtaining a swab, typically nasal, RNA extraction and conversion to DNA, amplification by PCR, and then result interpretation. RT-PCR can lead to indeterminate or negative results due to low viral loads or unsuitable collection, the timing of collection, handling of samples, and storage of swabs, or failure during the reaction process. Imagery techniques such as CT and X-ray can also be performed to aid in diagnosis. Further, antigen and antibody tests in conjunction with other diagnostic tests can be used to determine if an individual has been exposed to SARS-CoV-2. Droplet digital PCR is a uniquely suited molecular tool for diagnosis of SARS-CoV-2 infection that in addition to being a highly accurate and precise tool for frontline diagnosis, may help in situations where other molecular assays struggle due to some of the reasons I mentioned before. The Bio-Rad SARS-CoV-2 ddPCR kit is a powerful diagnostic tool in the battle against COVID-19 with sensitivity and precision beyond that of qPCR assays. This test is a partition-based endpoint RT-PCR test intended for the qualitative detection of nucleic acids from SARS-CoV-2 in nasopharyngeal, anterior nasal, and mid-turbinate nasal swab specimens, as well as nasopharyngeal wash or aspirate and nasal aspirate specimens from patients suspected of having COVID-19 by their healthcare provider. It targets the N1, N2, and human RPP30 genes and utilizes positive and negative controls from Exact Diagnostics. It has been shown to have high sensitivity and precision in low viral abundance samples, as well as resistance to an inhibition often seen in RT-PCR testing using traditional methods. The SARS-CoV-2 ddPCR workflow has 4 main steps. Preparation of the reaction, in which you combine your assay, RT, ddPCR supermix, and RNA samples or controls. Generation of droplets using 20,000 droplets per well on our automated droplet generator. Polymerase chain reaction using a one-step protocol for endpoint amplification, followed by reading and analysis of the droplets using easy-to-use software with integrated controls and other flexible tools. ddPCR detection of SARS-CoV-2 has several key advantages over other molecular methods. These include sensitivity and precision in low viral load samples. This minimizes indeterminate results. It is scalable to run 5 to 279 patient samples per day on a single platform with a modular design to increase throughput dramatically. Droplet digital PCR is highly precise in low viral load samples due to the partitioning that helps to increase the number of replicates per well. Lastly, this is a very flexible solution and easily amenable design to adapt to new needs in COVID-19 pandemic. For example, there has been shown to be utility in wastewater surveillance as a method of early detection outbreak. Earlier this year, a company named BioBot demonstrated that SARS-CoV-2 detection in Boston area sewage foreshadowed clinical case increases by as much as 10 days. Similar results were recently shown for the New Haven community in a report from researchers at Yale University, who found detection of the virus in sewage sludge was a 7-day leading indicator of positive testing and could provide 3 days of lead time on local increases in hospitalizations. ddPCR is showing great utility in this area and is rapidly being utilized for wastewater testing. Now let's move on to serological testing and the tools that Bio-Rad can offer. Currently, FDA recommends that labs utilize validated tests with EUA due to the poor performance of many tests that initially flooded the market. There are more than 30 EUA serology tests available that detect different antigens, including the spike protein and its receptor binding domain, as well as the nucleocapsid. These tests have been developed that utilize these antigens for IgM, IgG, and total antibody. Some data indicates that the nucleocapsid is sensitive though less specific, while the spike protein might be less specific but more sensitive, with the RBD also being more specific and less sensitive. But I feel that this is under debate, and combinations of individual spike protein or nucleocapsid have different performances dependent on the data that the manufacturers have put forth. Currently, there are not guidelines that indicate whether to test for IgG, IgM, IgG and IgM, or total antibody. However, some data indicates that total antibody may increase sensitivity. We will cover this shortly. It is important to minimize false positive test results by choosing an assay with high specificity and by testing populations and individuals with an elevated likelihood of previous exposure to SARS-CoV-2. Alternatively, an orthogonal testing algorithm employing 2 independent tests in sequence when the first test yields a positive result can be used when the expected positive predictive value of a single test is low. Antibodies most commonly become detectable 1 to 3 weeks after symptom onset, at which time evidence suggests that infectiousness likely is greatly decreased and that some degree of immunity from future infections may have developed. However, additional data are needed before modifying public health recommendations based on serologic test results, including decisions on discontinuing physical distancing and use of personal protective equipment. As we discussed earlier, early disease detection includes IgA/IgM, and then IgG detection extends for significantly longer and may be an indicator of long-term immunity. This chart gives an example of how IgM and IgG compare in a test individually and together, noted as the total antibody at different time points. Total antibody exhibits the highest % positivity at all stages and days of the disease progression, ranging from, on the left, 1 to 7 days, 8 to 14 days, 15 to 39 days, and overall. Bio-Rad has commercialized a total antibody kit called the Platelia SARS-CoV-2 Total Antibody Assay. It is an immunoassay kit to screen for total antibodies IgM, IgA, and IgG to SARS-CoV-2. It runs on an open microplate format. It can be run manual or fully automated, and it's for detection in human serum or plasma. It provides one total result in one test with all reagents required as part of the kit. And this current kit format is a 480 test kit, and the configuration is 5 plates times 96 wells. As I mentioned, the Platelia assay runs on an open microplate system, allowing a scalable solution to meet the lab's testing capacity. From low-cost, low-volume manual testing to high-volume robotic automation. While the assay is recommended for use in the Bio-Rad validated fully automated EVE system shown at right, or on standalone systems like those shown on the left, it can also be run on other validated manual or automated platforms. This means the platform meets a very wide range of volume and throughput needs. Let's take a look at some of the clinical data supporting the utility of this test. A multi-site evaluation was performed with samples from a general asymptomatic population of pre-epidemic individuals. This includes blood donors and hospitalized patients, and on samples from patients with clinical symptoms of coronavirus COVID-19 that tested positive with SARS-CoV-2 RT-PCR. Through clinical evaluation, the assay has demonstrated specificity of 99.3% with no cross-reactivity observed in specimens from patients with antibodies to non-SARS coronavirus or other medical conditions. For sensitivity, a longitudinal study was performed on 50 patients in the intensive care unit at 3 hospitals with clinical symptoms of COVID-19 and with a SARS-CoV-2 PCR positive result for a total of 127 samples. Of the 50 patients tested, 49 became positive. The patient that remained negative was not tested greater than 8 days post-onset of clinical symptoms. This led to an overall sensitivity of 98%. Earlier this summer, a publication in the Journal of Clinical Microbiology highlighted an early evaluation of an RUO version of the assay comparing its performance in clinical samples to 5 other commercial antibody tests. This RUO version was for IgM and IgG only, but still demonstrated greater than 91% sensitivity when utilized after 14 days and 100% after 21 days. And the authors found that Abbott, the affinity assay, and Bio-Rad to have the highest clinical sensitivity and specificity compared to the other EIAs. This is the chart showing this data, and highlighted here in the blue squared row is the data for the Bio-Rad total test. Again, showing that at greater than 21 days, 100% sensitivity and 100% specificity when looking at negative samples. So let's move on to another assay that Bio-Rad is in developing. This is, we are proud to introduce the BioPlex 2200 SARS-CoV-2 IgG panel. This is a multiplex assay for the qualitative and semi-quantitative detection of IgG class antibodies against the RBD, S1, S2, and nucleocapsid protein of the SARS-CoV-2 virus in human serum and plasma. The assay benefits include a composite result as well as a discriminatory result for each marker and a semi-quantitative result for individual markers. It is fully automated with a time to first result of 45 minutes and then one result every 36 seconds after that. Up to 100 samples an hour can be run. It comes in a 200 test kit format requiring just 5 microliters of sample. This includes calibrators with 5 calibrator levels per set, following a 4PL calibration curve, and 2 levels of controls per box. This assay is in development with EUA expected later this year. Early data from the development process demonstrates that there is no cross-reactivity with greater than 99.6% specificity in healthy human donors and greater than 95% specificity in infectious disease and autoimmune samples. There is no matrix effect seen for all targets when comparing serum versus plasma. A sensitivity study was performed on 105 samples with a confirmed SARS-CoV-2 PCR positive result. With specimens greater than 15 days after the onset of symptoms, 96.2% were reactive with the Bioplex 2200 IgG panel with highest sensitivity derived from RBD. It is worth noting that this assay is still under development and that the final performance characteristics are subject to change. In summary, Bio-Rad provides a wide range of products for use in the support of COVID-19 diagnosis and monitoring, confirmation of test results, and therapy or vaccine research and development. From molecular testing using real-time PCR or Droplet digital PCR to immunology assays, our wide range of solutions provide best-in-class sensitivity and accuracy. I thank you for your time and welcome any questions you might have.